haururu koru ta atoa. O te hakaro mai te aamu tōku hinua. Pei wa ana tōku aamu. He hau uatu I tuku ora.
I think the main reason to be here is how many of you have heard of the Rapamycin as a life-extending drug and that discovery being at the Health Science Center?
I've seen a little bit of it, yes.
So come on, let me see. I want to see.
Yeah,
no, never heard about.
Seen the mice footage. So I think
Yeah, exactly.
Well, put your hand up then. So the point is, I think a lot of people know about the story, but they might not know the details about how it came about and the person behind the idea. And then Dave is very generous with his attributions, I must say. And thus he will also point out the team effort that it took to make this happen. And then I think that the ITP program is utilized or underknown. Not very many people know about it. There's actually a call for applications right now for the ITP program. So I thought it was timely to do this because people find out how the ITP program works. If you have a fantastic idea of a drug that you'd like to test, if it's capable of extending lifespan in rodents, this would be the time to do it. And I'll be happy to send you, I guess, the link to that RFA or the request for applications that's going on right now. Oh, good. And then I joined Dave just when the 2009 paper came out, and I joined his group in order to do the cancer-specific study with Rapamycin. The original one was just any lifespan extension in mice that are wild type for any genes that we know of in a heterogeneous population. And I came in and started using mice that are susceptible to cancer and testing the effects of Rapamycin on it. So I guess Dave will start, and at the end I will have a couple of slides on asking for your input and a little bit about genomics and core facilities available at the Health Science Center. Again, more of resources to you and more ways we might be able to work together.
Okay, so this is a story about Rapamycin. Can we turn the lights down? So these are those statues from Easter Island there. And this is a picture from the astronomy picture of the day. And the photographer caught these statues as they're looking toward the Milky Way. And so us basic scientists, we're always looking to the stars, you know, for good things. And so I thought this was an appropriate picture to start with. And then this is a quote from Richard Feynman. And he was a very famous physicist. Quantum man, they called him, and he had this statement, and this is very, I think, apropos for my role and the role of everybody working here on Rapamycin. So let's get started. So we're going to start with a movie. So these are some mice here. They're representative from 3 different experimental groups. And so hey.
Come on in. Steve.
Have a seat, Daniel. Good to see you.
I'm supposed to introduce you.
So these are mice from 3 different experimental groups. And so I'm going to ask someone in the audience, which of these mice are the youngest? Just looking out. You have a guess? Mm-hmm. Can you see them okay? So would this be the youngest? Or these or these? These are youngest.
Yeah.
These guys right here look younger than these guys. Why is that? Because they're moving around good. They look better. This female up here has a big old tumor that you can see on that. So she's not going to last long. Okay. Well, actually, all these mice are about the same age. Okay. And so the question now becomes, what experimental protocol did we do to these 2 groups of mice here to result in these looking so much better at the same age as that one? Okay? And the answer is that this group over here was diet restricted, and that's a classic way of extending lifespan and health span, because these mice look healthier than these guys up here. So this has been known about for 30 years, that calorie restriction would do this. This group over here looks as good as these, and they've been treated with a drug we call rapamycin. We call it E-Rapa, and I'll tell you why we call it E-Rapa in a minute. Okay? So the subtitle, I guess, for this talk today is aging and cancer, and can we kill two birds with one drug? And that's kind of an audacious question, you know, that you could use one drug to attack something as pervasive as aging and also cancer, but it's not so outlandish, as you may see. Okay. So rapamycin, from the old perspective, is that in the 1970s is when this drug was discovered. It was discovered by a company, scientists working at a company in Canada, Aris, which later became White Aris, which now is part of Pfizer. But what they did is they went around the world collecting soil samples because they had figured out that the best source of antibiotics is soil. And so they collected soil from all over the place. And the sample from Easter Island, which goes by the name of Rapamycin according to the native, had a compound in it, which they named rapamycin. And it's a macrocyclic lactone. It's a typical looking antibiotic. It's actually made by bacteria in the soil, and it was developed first as a fungicide. Okay, and then they were testing some cells in culture and found out that immune cells seemed to be suppressed by this drug. And they said, whoops, we can't use it as a fungicide because it's going to inhibit the immune system, so they started marketing as a immunosuppressive. And today, in the clinic, that's used, Rapamycin is used in the clinic as an anti-rejection for transplants. It's also, in 1997, they discovered that cancer cells would be suppressed in growth. And so it's also used in anti-cancer applications today. And it's also included in drug-eluting stents. You want to get through?
Oh, sorry.
Well, you're not going to be able to.
Yeah, there's a computer monitor there.
You can see it right here, I guess. Okay. Now, if you want to stop me and ask questions, please. I'm not a formal person. So, the new perspective on the drug that I thought of about 10 or 12 years ago was that rapamycin could be a drug to combat aging and associated diseases. Okay, so this is some, gets a little technical, but a little bit of signaling will give you part of the rationale about why I thought this would work in a longevity application. So this is about TOR signaling. Target of rapamycin is what TOR stands for. It's in two complexes in a cell, this protein, and complex 1 and complex 2. TOR is one member of about 6 other kinases. They're called stress response kinases. And ATM, if you're familiar with those, ATR is another one. There's DNA-PK. There's several of these stress response kinases that this particular kinase is a family of. And so what it does, though, in a cell autonomous fashion, it also has non-cell autonomous functions, but the cell autonomous functions are to monitor those cells for inputs like growth hormone, IGF-1, insulin like growth factor, and other kinds of cytokines that signal through the cell, and those eventually impinge upon the TOR complex. And it's also then monitoring the energy state of the cell. It's also monitoring the nutrient state of the cell for and over here it's about amino acids. So it's amino acid sensing system, energy sensing, and also growth factor. So what it's doing, it's coordinating all those signals, and basically what this kinase does is it regulates cell growth. Now, I'm talking in mass, not in numbers. So before cells can divide, they have to grow, and this kinase regulates that process. The functions that, downstream functions are protein and lipid synthesis. So that's part of the cell growth part. It also promotes cell senescence. That's when cells start to poop out. And it also regulates energy metabolism. The other thing that it shuts down when all these things are sufficient in cells is autophagy and lysosome biogenesis. Both of these are involved in recycling components of cells for cell survival when times are bad. So under these conditions, you get a normal lifespan and all the attending, age-related diseases. So the interventions that have been shown to extend lifespan impinge upon this pathway. For example, diet restriction impinges upon the nutrient sensing part of this system. The energy state is also impinged upon by diet restriction. One of the things I didn't mention is, on top of all this, let's say a cell is radiated with radiation. Well, you wouldn't want a cell to grow while it's undergoing a stress like that. So this kinase, the TOR system, responds by shutting all this down until the cell is ready to go again. So it's a stress response kinase in this sense. So it's combining and integrating all these things. Okay, so if this is the system now that responds to diet restriction and growth factor restriction, the idea back then was that inhibition down here with something like rapamycin would mimic these other kinds of restrictions that have been shown to work. Okay, so that's what I proposed to the intervention testing program. And so a little bit about the intervention testing program. It's part of the NIA. It's sponsored by the NIA. There's 3 test sites that participate in this, and they each have a director. The director here in San Antonio is Randy Strong. Some of you may probably know him. I know you do. The other one lab is at Jackson Labs, and this is David Harrison. And then Rich Miller is the site director at Michigan. So each one of these places tests the compounds that are proposed to the intervention testing program.
Do you have to have a beard to get that job?
What's that?
You have to have a beard to get the job.
Yeah. So I was a sponsor for the rapamycin proposal, and it got accepted in 2005. And Randy and I started working, trying to figure out, could we feed mice rapamycin? Now, we had to feed them because rapamycin is insoluble. It can't be put in the drinking water. And so we did. We put rapamycin just, out of the bottle into some food and fed it to mice. And sure enough, we could get the expected effects on tissues based on the signaling system that we knew about. The problem was that most of it degraded. 80% of it would degrade, and rapamycin was fairly expensive. So it wouldn't have been possible to do the experiments financially. It would just cost too much. And I thought at the time, well, we're dead. Nobody's going to be able to do this. Well, Randy's a pharmacologist, and he figured out a way to stabilize the drug. And what he proposed was that if it was encapsulated in a material that would survive the food processing process, then perhaps it would be feasible. So he went to the Southwest Research Institute, who developed microencapsulating drugs. And sure enough, they had a suggestion that it would be a compound called Eudragit 100. That's a polymer that, a plastic, I think, a polymer that the drug can be encapsulated in. The mice would eat it, it would go through the stomach, it would survive the pH environment and be released in the intestine where when the pH approached neutral pH, okay? So it was a enterically delivered formulation of the drug. So that's why we started calling it E-Rapa. No one had ever done that before with a rapamycin compounds, and so targeted delivery we think is going to be an important aspect of this whole story. So that's what this is. It's a special enteric release formulation. And the problem was that the cohort of mice that were going to get rapamycin had gotten old, okay? And in fact, they were 20 months of age. And in human years, that's about 60 years old, okay? And so these were old mice when they started taking it. Everybody said, oh, it's not going to work. The mice won't. This is rapamycin. It's going to kill them. And so, but Dave Harrison, I think, is the one that said, well, if it did work, it'd be big news, okay? And so they agreed to start it. And the concentration of the drug in the food is 14 parts per million. The dose is 2.24 milligrams per kilogram per day. And that was based on some of our preliminary data that we'd gathered in coming up with the encapsulating process and all those experiments. The other thing to point out is that the ITP uses genetically heterogeneous mice. Okay? And the reason for that is if you use inbred mice, it's a possibility that whatever drug you're testing will cure that disease, and that'll be the reason for an extension of lifespan. And they're not interested in that. They're interested in overall aging. Okay, so I mentioned the 3 test sites. Any questions up there? Okay, so this is, I took this from the National Institutes of Aging website, and this is the intervention testing program. This was the first cohort of mice that they tested. This was the second one, and Rapa was, here's what the Rapa cohort. So this was in 2005. I just took a few more to give you a flavor of the kind of things that the ITP will test. So this is cohort 3, and they decided to do rapamycin starting at 9 months, and I'll show you the results of those data. And they also tested simvastatin and resveratrol. You've probably heard of resveratrol, right? It's the stuff in wine that everybody thinks will extend lifespan. So this is cohort 5. And in this one, the question was the original dose the best dose? And so they're testing 3 different doses. And I'll show you data on that too. Okay? Now, these are other compounds, and I don't know much about these. Okay, this is acarbose, which I think, y'all may know better about it, but I think it inhibits carbohydrate absorption. So this is kind of like a calorie restriction thing, I think. Methylene blue, I have no idea why that one.
It acts as a nitric oxide inhibitor, and it's involved with redox cycling.
There you go. Thank you. 17 alpha estradiol. Why would that? Anybody have?
Females live longer.
Well, this is not beta.
It's alpha. Right?
It's alpha. And I'll show you results on that. So this is cohort 7. And again, they're testing metformin. And metformin plus rapamycin. And let me just go back to this signaling thing. I'll show you the rationale there. So metformin actually will impinge upon the nutrient sensing part of this pathway. So that's the part of the rationale for that. And so they're going to also combine, I think this is Arlen Richardson's proposal, I'm not sure.
Arlen?
It was more Adam Salmon's proposal.
It is?
I can see what you're talking about, Dave, because
It's
I
over here.
there. I just don't get your
Oh, I didn't know
dynamic.
you were here.
Dynamic.
Okay, so this is the ITP, And so it's really
Oh,
good.
Dave?
Yeah.
The other reason we did the metformin was that it looked like the rapamycin mice might be insulin resistant.
Ah.
And so we
Overcome the yeah.
Yeah.
Okay.
And Dr. Zlaga has been doing experiments combining metformin and rapamycin as well, so a lot of people think that that might work.
Yeah. Okay. Okay, so I always like to show this slide. This is from the Nature paper because the fact that rapamycin would extend both males and female lifespan started at such a late age was really a very significant finding. And this shows when rapamycin was started. Right here are the arrows, and you see the extension in lifespan. Now, you'll notice that before the drug started, the males here were starting to creep out a little bit. And that's because at that time, the ITP hadn't settled on a diet. And each one of those places were using a different diet. And except when they put them on rapamycin, then they all use the same diet. So this is a diet difference that males can pick up there. It's just amazing. That didn't show up in the next one. They corrected their food thing. So I mentioned this a while ago with starting Rapa early help, and this is the cohort I told you about. And I just wanted to show you a little bit of data indicating that we see the expected reduction in inhibition of mTOR system by assaying something called ribosome protein S6, the phosphorylated state versus the non-phosphorylated state. So when Rapa inhibits the mTOR system, this goes down, and this just shows in adipose that we're getting nice reduction. So this was the results of the starting at 9 months. Just the arrow here in each one of them. And now you notice we don't have that difference in the beginning, and we're still getting an extension of maximum lifespan in both males and females. And I'll just mention, and Arlen can speak about this as well, that when you see a maximum extension of lifespan, what that means in fancy terms is that all competing causes of mortality are inhibited. My way of saying that is most age-related diseases are inhibited, so that you get a maximum extension of lifespan. So a lot of people wonder, did starting at 9 months, is that any better than 20 months? And it's not much. In males, it's no difference. In females, there may be a little bit of a difference here. The blue one is the 9-month group. It wasn't statistically significant, I don't think, but there is a little bit of a bump there. Okay, so basically what that means now is that you don't have to start an anti-aging intervention while you're young. You can wait till you're older and still derive some benefit from it. So again, is that dose the best one? So that cohort that I mentioned a while ago is still in progress. So they're getting food at 4 parts per million, 14 parts per million, and 40 parts per million. Approximately 3 times less and 3 times more. And we had some mice to assay, some monitor mice, to see if rapamycin was having, we could see any dose response in the pharmacodynamics. And so this is Marty Javer's data here. This is from blood, liver, and fat. And so nothing was detected on the low dose. This is the medium dose and the high dose. And you can see getting pretty good blood levels, 30 nanograms per mil. And way out here, this is really a lot of rapamycin, okay? And this is picograms now the green one is fat and the red one is liver. So you can see we're getting picogram levels of the drug in the expected organs. Now, the first organ that sees the drug after it goes through the intestine is liver. So I thought maybe the liver would have a higher one, but maybe it's metabolizing it. So anyway, that's that. And I'll just go through this. So again, we see the expected sort of dose response and the inhibition of this ribosomal protein S6. This is the phosphorylation site right here, going down. Nice statistical significance, and liver and muscle seems to be really nicely affected. Okay. That's really responsive. Skeletal muscle, that's the gastrocnemius. So, higher dose. What do you think? Is it going to make them live longer or not?
Kill him.
Huh?
It'll kill him.
It'll kill
It's toxic, right?
He knows. What do y'all think?
Well, don't tell him he knows, otherwise you're gonna hear.
Well, it's amazing. So this is females. You probably hadn't seen this, Earl.
I have seen, yeah, I think I have, yeah.
This
But
is,
not
I
quite
only
that
got it,
far out.
I only got it yesterday.
Not that far out. It was 2 months earlier.
So that's a few months old. So here are females, and this is sort of complicated because it's got a lot of compounds in it. But so all these grouped together right here, that's control under there. And in males, there's that acarbose thing, and also the estrogen. So they're sort of in this group right here. And in females, there's the low dose. So we're getting a really nice extension of lifespan with the low dose even.
Can you give me the colors?
That red.
Okay.
It's a red one right here.
Okay.
Okay, the blue one's a middle dose, and that's the high dose. High dose, middle dose. And that's just incredible.
They may never die.
So Rich Miller believes now that this is the high dose is going to equal calorie restriction and its ability to extend lifespan. And nothing's ever done that before.
And they were started at 4 months now, or at
9
9
months.
months? 9 months.
It looks like it's more than caloric restriction.
That's just what he said in the email thing. So, okay, so that's that. The males now don't have as, they still respond in a dose response manner, but it's not as good as females. And that's something we see, that females seem to do better on Rapa than males do.
But estrogen is working, right?
No, it's acarbose that's working. And estrogen works a little bit.
Yeah.
Okay? So this is estrogen down here. The low dose in Rapa doesn't do anything in male. That's the red one. And the high dose does, the green one right here. So the mid and high dose are working in a dose response manner.
So are they going to combine estrogen and rapamycin in males?
I don't know. This is acarbose and acarbose thing. So that works in males, but it didn't work out. So figure that out.
The rats haven't been noticed.
Hmm?
No, rats.
Hmm?
Or mice. Do they have menopause? They don't.
I can't understand this.
He was asking about menopause and
Oh, no.
No. So old female mice are not menopausal.
Right. So one explanation, it's a trivial explanation, is that mice metabolize rapamycin faster than females. It's a suspicion we're getting, okay? And so this may be trivial in that they just have higher blood levels or tissue levels or whatever. And all these differences here, low, medium, high, are all statistically significant at this point. In males, it's just the mid and high dose.
Or is it because of the fat tissue?
Hmm?
There's a higher concentration of fat tissue. Females have a higher.
Is that right?
I'm just.
Yeah, I don't know. We haven't looked at that. Okay. So, well, this is out of line. I pushed the wrong way. Sorry. So, a question for this group is, that I thought might be interesting is, does ERAPA slow aging? So that's what we're really interested in. We're not really interested in keep them alive. We're interested in having a better quality of life. And so these are some what the ITP calls age-sensitive traits, and this is at 22 months old. They're measuring these, and these are in the het mice that are in the ITP protocol. So they looked at liver degeneration. So this is what you see in a young mouse, and this is what Rapa prevents that. Myocardia nuclear atypia. I'm not sure exactly what that's all about in terms of aging, but Rapa also seems to inhibit that. Endometrial hyperplasia. Rapa does a nice job of keeping that down. And adrenal tumors. I'll talk more about adrenal tumors in a minute, but this is also nice. Here's more. Adrenal hyperplasia is not affected. I can't say that word. Something going on in the adrenal gland. Telangiectasia, blood vessels forming, right? That's prevented a little bit. Ovarian cysts are prevented. The thyroid cold follicles, that means I think ones that aren't making thyroid hormone are also affected. Lung tumors are prevented. Spontaneous activity, mice start to slow down like I showed you in the slide, and Rapa, prevents that. So what they found though, one negative effect was cataracts, and so old mice apparently get cataracts, and Rapa sort of potentiates that. So that's not a good side effect. Both males and females. As mice get older, their testes starts to degenerate. Rapa promotes that degeneration, and that's something that's been noted by other people here, like Chris Walter, I think, has noticed that in testes, correct?
And it reduces circulating levels of testosterone.
Right.
And we observe that very easily because when the mice are on E-Rapa, the males, they stop fighting. So you immediately get unblinded to the diet that they're on in the cancer models. And obviously they get very upset when they're injected with tumors and the tumors start growing. So that usually makes them more aggressive towards each other, and they fight, and there's problems, and the E-Rapa makes them nice and calm and easy. But that makes it very difficult to analyze the two groups, because this group is really healthy and this group is all beat up, and you can't study males. So we've been studying females mostly.
Okay, so I told you we were going to talk about cancer, and real quickly, this is stuff you guys don't need. We're an aging population now, pretty much. In 2009, there were 1.5 million cancer cases. 500,000 people died from it. It's going to get worse. There's a doubling of the greater than 65-year-olds up to 72 million by 2030. That's about 20% of the population. And the age-adjusted mortality rate for cancer in 65-year-olds is 15 times greater. So this is going to be a big problem for the aging population. There's also a shift in the risk-benefit ratio of the anti-cancer drugs that you get. So that's another. The NCI has finally recognized that aging is probably something they should pay attention to. Like, this was a provocative question. And it says, does our understanding of the process of aging help explain aging carcinogenesis as a function of age? Well, duh, it's only been known for 30 years. So, to me, the big question is about cancer is if we slow aging, like I've shown you can happen, can cancer be prevented, delayed, or reduced in severity? The one that's not addressed is does that affect the tolerance for these anti-cancer therapies, these nasty drugs they get. So I mentioned previously that diet restriction is a classic form of longevity promotion. Growth factor restriction, these are the dwarf mice that they live a long time. They're about half the size and they live about twice as long. And so somehow growth factor restriction and diet restriction also. And it's been Yuji Aquino here is the person that has looked at cancer in both of these dwarf mouse models, and there's a reduced incidence and delayed occurrence of neoplasia in these mice. So delay aging, you delay cancer. And diet restriction's been studied 30 years, and these are mostly studies that had to do with, chemical carcinogens or radiation or something, and calorie restriction was retarded in both sets of settings. Spontaneous cancers have also been documented. One of the recent ones being the rhesus monkey study that took 20 years, and it was published in Science. So that's the history. So let's go back to the 9-month cohort and look at some of the cancers. And they looked at the end of life. As you can see here, cancer is the thing that kills both these mice, and there was no difference in the numbers in control versus Rapa. But what they did notice is the mean age of death for control versus Rapa was extended for liver, lung, and mammary carcinomas. Okay? That wasn't true for lymphoma and hemangiosarcomas, which are the other big killers for these mice. So somehow Rapa might be having a differential effect, and was one of the reasons that we were interested to do this study here. So this is the RC2 grant. Arlen Richardson and I put in this GO money that was several years ago, and this grant grew out of something called the Rapa Roundups, where we got people together and talked about potential projects that Rapa would have an effect on. And there were 9 projects that were funded, I believe, Arlen, right?
Yep.
And they range from diabetic nephropathy with Kazanov, radioisomate had one on atherosclerosis. There were 3 immunology projects that people looked at. There was a Parkinson's project and one on autophagy, and I can't remember the other ones. I'm missing
2 on Alzheimer's.
2 on Alzheimer's. That's right. The big ones. So I'll just say that our story here is just one of many stories. Dean Kellogg right here is working on Rapa too, and some of his projects. So this all spurred a lot of activity here in terms of research. This project was the effect of Rapa on cancer. So I'm going to try to go through this. I'm running along, right?
It's okay. I'll figure out when I do one.
Okay. Okay, so this is a model here that we proposed, and you'll see part of the rationale in a minute, but let me explain to you first. This is the RB model. This is a model for retinoblastoma. This mouse was constructed many, many years ago in the 90s to mimic a human condition, childhood disease that they get in the retinoblastomas. Mice don't get retinoblastomas, they get other types of tumors. But retinoblastoma is a protein that regulates the cell cycle. It also regulates cell differentiation. It's mutated in many, many types of cancers. It's the prototypical tumor suppressor. So when it's working well, you don't get cancer. When it's not. And so this mouse had 1 good copy and 1 bad copy. If it's 2 bad copies, it's embryonically lethal. Okay. So this is a mouse that's highly prone to cancer. They're synchronous in their development. So we know exactly when they're going to form. We know where they're going to form. And so these tumors that form are neuroendocrine type tumors in the pituitary, thyroid, and adrenal. Okay, it's 100% penetrant, which means all these mice are going to die. So and they die pretty quick, which is another reason we thought they'd be good for the GO projects, because we only had 2 years of funding. And it's gone on much longer than that. But okay, so this is Alex Nicotin, and just to give you a sense of how these form, at about the time of birth, in a pituitary gland or adrenal gland or a thyroid gland, there's a loss of that 1 good copy, that's the red cell. It has now a proliferative advantage. I would say also it now has a growth advantage in terms of, as I'll propose in a minute. And that tumor will grow, and if it's thyroid, it can metastasize to the lung. And I'll show you some data on that. So, one of the interesting things was that I worked with the aging group here, Jim Nelson primarily, and Arlen, and we did a diet restriction on this model. And one of the interesting things is that calorie restriction has no effect on this model in terms of growth of tumors or lifespan. And that was actually you know, a negative finding, but to me it was a discovery, because now this is one potentially important aspect of RB. Is RB necessary for the effect of calorie restriction? It was a negative finding, and nobody would ever fund anything to further to study that. So this is the blood levels. We started feeding these mice the Rapamycin, the medium dose. Okay, these are the blood levels. Not as good as others, so it's about around 4 or 5 and around 4 nanogram per mil. But we were getting nice, acceptable blood levels. And so the question to the audience is, do you think Rapa will mimic calorie restriction or not?
Yes. I would say yes too.
No, I'll go the opposite, just to be contrarian.
And you get the prize. It's really amazing. So this is males, this is Rapa, this is control. Females, Rapa, controls. Can you see that, Arlen?
Yep. Oh.
That's a really potent extension of lifespan.
And I would point out is the levels that you see, you said the Rapa levels
Yeah.
were low.
Yeah.
In general, I think there's something about those het mice
Yeah.
that they metabolize because they have much higher levels.
Yeah.
In other words, I think all the mice that we did in the GLOW study that were not, because they weren't het mice,
Yeah.
they were some background of black 6 or
Yeah.
some other inbred strain. Almost all the levels are around 4 or 5.
Correct.
Arlen, remember, we bred these, so these are not the funky mice. These are C57 black 6.
But even the funky mice
Yeah.
were this.
Oh.
And Rito's mice were even lower.
Yeah.
So all of the Rapa levels that sometimes you get into them saying, well, the Rapa levels that Rich Miller and those guys published were high for humans. The values that we have here of blood levels are roughly the same blood levels that you see in patients that are treated for cancer monotherapy.
Can you judge, though, the effect of your different blood levels based on like S6 kinase phosphorylation levels? Trying to standardize
I don't know
it?
whether we've ever done that. All I think is that I think that for example, when we did the late life study in black 6 mice, we saw just a very faint increase, nothing like they saw in the in the 4-way cross. But if we do it earlier, we see a very robust effect. So I think that if we would go up to higher levels in the black 6, we'd get better extension. In fact, you might even get better extension there,
Yeah.
Dave.
Oh, I'm sure we will. The other thing is C57 black 6 have a SNP mutation that affects mTOR regulation. So mTOR itself is mutated in C57 black 6.
Really?
It is part of the 3-way cross, of the 4-way cross that they use, and it is likely that when you do genotyping of the mice that die sooner may very well have the contribution from the C57 black 6 strain. And I proposed that a few years ago, but Rich Miller was already on top of it, and he already had plans to genotype all the mice. So I think they're supposed to be doing that now. I know, it's one of those fluke things in science. The strain that we pick has a mutation in mTOR. It's par for the course. I sent you the information, Arlen.
Okay, so the question was, did it affect the tumor growth? And so because these tumors are synchronous, I was telling Carolina about this, and she said, wow, we could do MRI imaging. And she went out and found some money. I can't remember exactly where it came from.
Very important. It's the IIMS CTSA grant, funded here. Very important to say that. And it was a translational technology resource, one of those small TTR grants that we submitted and got funded, that funded this work.
So I don't know if you can see, there's a little one, there's a bigger one, and there's a much bigger one. It's amazing to me that mice walk around with this size tumor in their head. But anyway, so Ruan Hardman in radiology did the measurements on these blind, and the tumor volumes were calculated by Carolina, and so these are the Rapas groups here, and the blue one's down here.
No, it's the other way
I'm
around.
sorry, the other way around. Utridrit here, Rapa here. I'm sorry. So it's definitely inhibiting tumor growth in the pituitary. Are we going to do thyroid too or not? No?
We did, remember we have the results back, and it's also reduced, but it's harder to always measure it, so we have a lot of missing data.
Now the other thing that I was really interested to know is what effect this would have on lung metastasis. And so the pathologist Yuji Aquino and Gene Hubbard are the pathologists in the Barshop Institute, and they did look at
Dave, I'm sorry, I want to, so going back to that previous, just thinking as an endocrinologist, do you know that those tumors make growth hormone? Because it, acromegaly is very hard to treat, so
no,
finding something like this.
They don't make growth hormone. We know that. They make, it's the corticosteroid pituitary cells that become tumors. Or in the intermediate lobe in the mouse, it's the melanotropic tumors. And C cells are the types of tumors in the thyroid. Adrenal, I think it's pheochromocytomas.
By doing the MRI, we were able to see that really it's not simply making the mouse live longer with a bigger tumor. It's also reducing increasing the time it takes for the tumor to get to that size.
Yeah. Okay, so I haven't talked about these data, so help me out, Carolina. Okay, so this is the grade that the pathologist assigned. 0 means they didn't have one, didn't have a metastasis. And Utridrit had about 12, and Erapa was 16. Okay, grade 1, Utridrit only had one of those, and the Erapa groups had more of those.
So the grades is based on the percentage of the slide that contains metastatic cells. So of course it's a scale, and it was 2 pathologists grading, and they took the means of blind. They took the means of 3 slices. They were not very far apart though, so it's not like they did the entire lung. So it was Chu's lungs. I think 3 slices per lung, if that makes more sense. So 4 would have a ton of them, 3 would have a significant percentage, and 2 would be an intermediate level, which is where the majority of the mice lie.
So this grade 2 here, there was a lot more in the Utridrit group versus the Rapa group. And so the bottom line is we think that it's having a positive effect on preventing, to a certain extent, the metastasis of these tumors.
And we're sending the slides to the Hyperion machine where it will actually calculate absolute percentages.
So that'll be a big deal. Okay, so I want to just, you hadn't seen this one, Carolina. So whenever I sent a draft of this paper to our co-author, Paul Hasty, and he thought and I mentioned the Menin story in it, and he thought this was more interesting, so I thought I'd just throw it in here today. So the RB plus minus mice have a multiple endocrine neoplasia type 1 phenotype. Okay, it's not perfect. They don't get prostate cancer, but they do get the other types of endocrine tumors like that. And so this MEN1 gene is involved in that. And when it's mutated, you get the multiple endocrine phenotype that is seen in humans. So this menin is a tumor suppressor, kind of like RB. And it has many functions. It's still being studied in terms of function. One, it mediates apoptosis via caspase induction. The other, it enhances transcription of the CDK inhibitors, P18 and P27. Okay? And it also inactivates RB1, and this is work from Alex Nikitin's lab. So when RB gets phosphorylated on serine 807, it's downregulated. So our data, do I need to stop it?
No, you
Go
could.
ahead.
our data indicate that mTOR is also a factor in this pathway. So this would be the model. Wow, I'm moving a little.
Yeah, it's one.
They're getting
Well, you've got till
started.
1:15 if you want.
Huh?
You got another 15 minutes if you'd like, and Dr. Musi owns the
Okay.
room, so you can go forever if he says so.
So this is what's observed in MIN1. And when MIN1 is inactivated, you get an inhibition of RB1. And the hypothesis would be that that is an activation of the TOR system. And when you get an activated TOR system, that's classic for tumor promotion, okay? And then the other way you can get it is, and RB is downstream of MIN. That's clear from Alex's work. And so, but when you get a mutation of RB1, you also get an activation, and that gives you a little bit different phenotype, but the same type tumor. And ERAPA, of course, is effective, would be effective in both of those situations. What you don't observe in any of the tumors, so if you cross RB and MIN1 plus minus mice, that's a double cross, you never see tumors that have both mutations. You only see them with this one or that one. And the idea is that when both of them are out, now somehow mTOR becomes inhibited and you have an apoptosis. So that's a question right now that this research sort of opens up. Okay, so that's that tumor suppressor. I'll try to go faster. This is the other one that we're excited about. This is a model for intestinal polyposis. It's called FAP, familial adenomatous polyposis. It's a hereditary condition. There's thousands of these polyps in a cancer, and it's due to a mutation of the APC gene. So this leads to deregulated beta-catenin. So these are beta-catenin-driven tumors that form in the intestinal tract of these mice. And humans, they get thousands of these polyps, and if the colon isn't removed, they will die from colon cancer. So this is a pretty aggressive type of cancer. But this APC man mice, plus mouse, is a model for these types of cancers. And they've developed numerous intestinal polyps. They can progress if the mouse lives long enough. Calorie restriction actually reduced those polyps. And so we wanted to know if this enteric formulation of ERAPA would be effective in this model intervening. And boy, the answer is yes. So this is the control mice. They die at 180 days. We're out past 600 days now. And 7 out of the 10 mice are still living, still going. Okay? So this is really an amazing result.
Yes.
Are the control mice calorie restricted?
No,
No.
not this one.
How much did calorie restriction?
They didn't do the lifespan. They just looked at the
polyps.
polyps. But this is a really remarkable result. Okay? And so, Carolina, maybe we'll have time, and you can mention the clinical stuff we're thinking about. So, to give you a feel for this, so this is the lifespan of C57 black 6. Paul Hasty put this on, and this is where our mice are dying on the high dose and low dose. Okay, so it's giving them a normal lifespan. Okay. So the thing that these mice die from is anemia, and so this is the hematocrit of the controls when they were starting to die, and you can see it's a very poor hematocrit. Their organs are real pale, and this is the Rapamycin at that time, and this was on August 18th in 2011. They had a normal hematocrit. And so we did the hematocrits again, and so these blue ones right here, this was in 2012, a couple months ago, and these are the blue ones. You can see they're starting to get a pretty poor hematocrit, but the high dose is still very good, okay, normal range. So this is disease-free survival, normal lifespan.
And this is probably so they're bleeding from the polyps?
Mm-hmm.
Yes.
And you can see when you kill them and bleed them, the blood is water as opposed to viscous when they are
Okay, so one of the questions is the mechanism, and we do something called polysome profiles, and this is in the intestine, and this is a way of assaying the mTOR system, and basically when you see a reduction of the ADS peak right here in this profile, it's a reduction of mTOR activity. And also these polysome peaks out here are all reduced. So that means, what that means in terms of what's going on in the tissues is that translation is really being inhibited quite strong. We also wanted to know in the colon, so this was a short-term treatment, and we got a reduction in the polysome, but a longer-term treatment reduced everything. So this is, we know that this drug is working in the intestine and the colon of the mice. Okay.
Why don't you skip the P53 story?
Let me just skip
I'm
this.
sorry, so what you showed before, this is instead of doing like a Western blot
Yeah.
or acid
It's a
kinase? Or
polysome profile.
Yeah.
So it's how much how much the ribosomes are bound to mRNA, translating them, and then also how much ribosomes are around, because we know that rapamycin reduces ribogenesis.
Oh, I
itself.
got it, I got it.
So it's both. And then now what we're proposing to do is to actually, as opposed to doing a quantitative how much of it moves away from translation, actually sequence or doing arrays to see which messages move from translated to not as translated. Does that make sense? So it can be specific types of messages that have specific signals, leaders that move preferentially from translation.
Okay, so real quick, P53, diet restriction works in P53 mice. So we wanted to know if it worked in ERAPA would work.
And these guys get with breast and colon? Cancer?
No, they're lymphomas, hemangiosarcomas, all the lomas, whatever they are.
Mostly lymphoma. Like 80% is lymphoma.
So this is minus P53 mice, no effect, males or females.
And remember that lymphoma was not reduced in the ITP trial either, so this should not be surprising.
You put back one copy of P53 and now we see the line starting to go on the right side. This is actually statistically significant. So this is saying the ability of ERAPA to extend longevity is dependent upon P53 being here. And of course, whenever you put both copies of P53 back in the same frame, you see that. Okay, so I'm going to skip all this.
So what was that last, the other one? So in this one,
plus, so this is like, well, this is...
okay, this is wild type.
Yeah.
Okay,
So we know it does
Okay,
it, but,
I got you.
but we had to do it, and this is I remember debating in our group meeting, should we do the plus minus, should we do it? So it's so expensive, you know, that if we hadn't done it, we wouldn't have known this, and so there's a grant in this right here, okay?
So are these RB mice, are they black 6? What background are they?
Yes.
Okay.
And the APC as well, they were generated as 129s and then back crossed at least 7 generations to black 6. The P53 are a little more mixed, but they're almost pure C57 black 6.
So I thought I heard that if you used higher levels of rapamycin, you could have an effect.
So that's
on
what
these
so that was my question here.
Okay.
Would a higher dose work better?
Okay. Oh, so you haven't done that?
We haven't done that.
Okay.
No.
But we do have RB mice, and we have to decide if we're going to keep the colony and, you know, and actually test the high dose. And I think we should, because it wouldn't take very many mice to do it.
Okay, so it works good in APC mice, it works well in RB mice. It doesn't work in minus, it may be dependent on P53. I think, Caroline, that's it. So I just have to...
Do you have my PowerPoint open?
Yeah.
And how many people need to go right now?
Mice seem to really like this group. Will people...
I don't have very many slides. I'd like to show the first couple.
I want to say this,
Okay,
Caroline. This wouldn't have ever happened if it hadn't been for this group right here. Okay? This is, and Arlen and everybody's been really great. Okay, so you want,
That PowerPoint is up to you.
the power.
Yeah, CVV, Rampa, GG, but is that not the PowerPoint, okay, PowerPoint's fine, isn't it?
I'm sorry, it's wrong.
So I'm only going to go over the first probably 6 slides since it's very late, but I'd like to discuss with this group any ideas you might have for patient populations that could benefit potentially from a rapamycin or E-Rapa trial. And I ran across a paper, so let me back up a little bit. So I'm a genomics person, so I'm actually the gene expression director for the genomics course, so if you want to figure out how to measure either RNA or DNA, you can come talk to me or send me an email. I'm also the co-director for the Core for Advanced Translational Technologies, which is supposed to be a core where we process samples for you. It's a turnkey operation. If you put in a protocol and you have an IRB approved, we can collect blood samples, we can process the blood samples, we can process tissues, and we have several pieces of equipment to do analytes, including a MiSeq for next generation sequencing, a Biomark for microfluidic PCR, and a Pyromark for pyrosequencing, and a Luminex instrument that's a FlexMap 3D instrument that can do many analytes with small amounts of volume. So if you're interested in any particular research project and you'd like to know what resources are available, we're happy to try to help you.
Pyrosequence, what is
MiSeq. And obviously there's also a HiSeq across the way with the GCCRI, and we work with them as well. At the Genomics Core, we also have an ion torrent, which is the pH-based sequencing. So pretty much, and if we don't have it in-house, we will set up a service with another institution. And the advantage of that is you can make sure that the samples you're collecting is what you need, the quality control is done here, and since we speak the same language as people in other genomics cores, hopefully we can facilitate that process for you. So it's just very hard, as you probably have experience if you go outside of the university, to get exactly the service you want and also figure out how to do the data analysis when it comes back. So if we don't have it here, we'll figure out who has it and how to set that up.
Do you have next gen?
Yes, we have a HiSeq from Illumina, a MiSeq from Illumina, as well as an ion torrent from Life Technologies, and a pyrosequencer from QIAGEN. So we're pretty much covered in that. We don't have a nanopore sequencer yet, but we might have one within a year. As you know, they're not commercially available yet. But yes, it's all in-house and running. Okay. So the reason I wanted to talk to your group, though, is translating our E-Rapa findings. So I've been talking to a lot of clinicians, and from the APC-Min results, we discussed this with Dr. Raul Ramos, for example, and Dr. Parker, who see FAP patients, and we propose to initiate E-Rapa treatment of FAP patients. The main finding I want to discuss with you is when I was looking at gene expression data that I collected actually in collaboration with Arlen Richardson, some of it was our arrays, and Arlen shared his data and I shared my data. It was a great way to double up our efforts. I noticed that P450 proteins, and which are encoded by CYP genes, were often popped up in our analyses, and these are genes involved in drug metabolism. And I wondered if this might have an effect on pain management and how opioids are managed, and if rapamycin might change that relationship between drug metabolism and pain management. So I went to Dr. Kenneth Hargraves, and of course he is the Mr. CYP. I mean, it's like CYPs are us type guy. And he said that, yeah, that's a great idea. I really think that might work out. So that was about a year and a half ago. And then I ran across this paper where they actually published that rapamycin reduced pain responses in a particular model for pain with 3 different assays. So one, the one that is used most often by Kenneth Hargraves is thermal hyperalgesia. They put the paw of the mouse on a hot plate basically, and the temperature at which the mouse or rat withdraws the paw indicates how sensitive it is peripherally. So as you can see here, it takes more time for rapamycin, for the mice on rapamycin to withdraw their paw. So once you do this as a hyperalgesic index, then it shows that Rapa significantly reduces this. And in this case, it's a tactile allodynia, so you use Fire's filaments. It's just a filament that applies a fixed amount of force that you can measure. And so with rapamycin, you need more grams of force before they actually react to it. And that's also a statistically significant reduction. So since a lot of people work in the palliative or hospice or, you know, in the elderly pain management and cancer pain management is a big issue, we wanted to discuss if there might be a patient population that gets rapamycin for a different treatment group where you might be able to do an observational study to see if we see this. Obviously, requesting the IRB to allow you to do Fire filament testing is relatively non-invasive, and it actually there's several papers published where it's not very difficult to do. Or potentially even an intervention situation where we might find a population where we can give eRAPA to do it. And I know Dr. Musi has a lot of experience with muscle biopsies and doing exercise regimens, and I think there would be a great expertise here to guide us in doing these types of interventions. And as you saw, muscle is very responsive to rapamycin, so it would be a great way to actually follow the biological rapamycin response in tissues and be able to correlate actual responses and drug doses, which might be different between patients, with the outcomes in pain management. So that was the primary thing I wanted to...
Dean, do you do any pain assessments?
We did. A decade ago, we were using thermal pain. This is just with rapamycin. They altered the thermal by itself.
And the reason I want to show this is they think that the mechanism is local translation. Remember the polysome profiles where we show that translation is inhibited? So rapamycin would be reducing the local translation of proteins that lead to increased excitability. So literally, for example, the capsaicin, where capsaicin would lead to increased insensitivity to pain, for example, by reducing the expression of those proteins that are induced during inflammation, etc., you might actually reduce the ability of the patient to perceive pain. So this is obviously possibly a mechanism where you do not develop any sort of resistance to the treatment or dependencies, because it actually blocks the protein that allows you to sense the pain itself, as opposed to binding to it and leading to requirement of escalations for pain management. I'm sorry, I'm not too much of a pain person, but I'm hoping that I make sense, and I'm showing you some preliminary data indicating that this might be a place to go. And again, because a lot of these assays are very non-invasive, I think it's a real possibility for patients that are already on rapamycin. And since in a lot of cases there is not much you can do for these patients, and given the toxicity data that we showed you, which is even at the higher dose, the side effect to mice is they live longer and happier and healthier, I think that I might be able to argue my way through IRB to allow us to do
with
this.
With the capsaicin, you can use it topically and you eventually get the analgesia. Would you expect the same with topical rapamycin?
I've never thought of that. I only consider, but this was actually thought topical. This was intracatheter injection. So absolutely, it is very likely that you would not need it systemically, although what I would argue is that rapamycin effects as an anti-inflammatory might be beneficial overall for the patient, and if the patient has cancer, the side effect of giving it systemically might be a reduction in their tumor. So it might argue for a combination of systemic, and that's what we did with the FAP argument, which is obviously we could try to just give it, you know, in the intestine, or do bolus injections, or get it only to the colon to reduce absorption. But because we actually think the systemic effects are beneficial as well, you know, we think it's better to just give it systemically.
Well, you could tell them about your protocol.
We're doing some stuff with topical rapamycin to try. The questions had to do with the endothelial dysfunction it induces. You know, here's a drug that's supposed to block atherosclerosis, but it increases superoxide production, it decreases nitric oxide production, it does all these things you wouldn't think would be good.
Your data indicates it's okay.
So, is that in which data?
Yeah, when he presented his seminar, in some cases, the oxidative stress, the oxidative went up, but they still improved in terms of their mitochondrial
The
function.
exercise.
Oh, okay, well, this was some studies done by cardiologists interested in the endothelial dysfunction aspects of it, and it increased, it was pretty clear it increased ROS generation and decreased endothelial responsiveness. So, but a decade ago or more, we used capsaicin, and we were looking at the peripheral effects of it, and so that's why we could use the thermal responsiveness. It altered it. The perception of thermal pain by 5 degrees C or more. So it's just wondered if the topical Rapa would do the same thing.
That's a great idea, because it would be much easier to justify.
Yeah, it'd be
and
easy
to get,
to do.
It would be perfect for preliminary data in order to justify proceeding. But I've heard Ken talk many times, and we've done some arrays together with his rat models. So I've worked with him scientifically, and he was enthusiastic about it, and it is such an enormous problem in the clinic, and in some cases you get to the point where there isn't much you can do. But again, in my case, I'm interested in looking at the translation effects. If in fact inhibiting translation of particular molecules in neurons is having an effect, and of course, I'm looking for collaborators with people such as yourself that have done this type of work. And then I can take, for example, cultures. I know Jim Leckleiters grows neurons, and then maybe we can do something in vitro and then do the polysome profiling. So
you might be able to look at what, just do some small skin biopsies.
That would be great. Actually, skin biopsies is what Dr. Monica Mita used to monitor the rapamycin responses in her sarcoma patients. So I have a protocol for that. I have IRB to go by to resubmit, and it was
that's what I'm working on
very
now.
safe
to do that exactly.
Yeah, and it was not very difficult. Also, the beauty of this is now you require less and less material to do your assay. So
I don't know whether you can do these polysome profiles.
Yeah,
you can. We
can, and yes, we can. It's not much, but we can get away with it because you don't need a very large punch biopsy for this anymore. Do with a smaller one. I mean, it's not the most comfortable thing, Dave, but after I saw the amount of material that you could get from the muscle biopsies, because that was a question during the seminar, I got really enthusiastic. Actually, it was Dr. Lucy's seminar that went, really got me thinking about this is doable. He has done all that. He has been able to measure this, and these were healthy individuals, and it was an exercise intervention, but still, I felt that this was really feasible and started thinking about it. And contacted Dr. Hernandez, and I think he contacted you for the exercise physiology. So we're very, and I do work with Dr. Thompson, so I am city trained, and I have submitted IRBs before. So I'm not completely naive about what this takes, but with the CAT core, it means that we have technicians available at any time to receive and process the sample. So the inertia to get something going is much smaller because I don't have to have somebody in place to receive them. I can train them within the core, and we have 4 staff members ready to take in samples and process, during business hours. Anyway, here's some information on the core facilities, and I'd like to point out there's a lot of data out there that's in gene expression that other people have collected, and it can answer some of your scientific questions. So if you have...
This lab is at the STRF?
The CAT is at the STRF, and then the genomics core is here in the 5th floor of the medical school. So I'm at both. And I try to put the hats on such that, I explain to the user what's available, and the user, we pick together what the best technology is. And there are very few pieces of equipment that are completely a conflict of interest. There's 2. The bioanalyzer is available at both, but in most cases we agree as a group as to which one is better for the user. But you're right, the CAT is at the STRF, just across the way from the MARC, and away from the CTRC. So this has really helped us in terms of sample collection and processing, because we don't have to do the shuttling with a car to main campus anymore. So this has been a big upswing for us. So there's lots of data available on the internet, and if you have a particular work and a particular molecular question, I highly recommend you see if somebody else has done it before. And I guess I'll present how does
CR and rapamycin work, the work that I presented before, and Arlen is a part of at some other time. Okay, thank you.
Thank you.
By the way, this is our story. There's a lot of Rapa stories around here. The Alzheimer's story is really amazing. Y'all should hear that sometime. Radioasmus has a good story too, and Kasi Tajna. So I'd recommend all those.
Thanks. Thank you.
Mhm.